Thank you. Well, the subject is very simple. I think we all know that the resolution of a light microscope, at least that's what we've been told at school, is fundamentally limited by diffraction. Now, at the end of the 19th century, this man that you can see here realized that the resolution of a light microscope is limited to a value which is given by the wavelengths divided by twice the numerical aperture of the objective lens. What does this mean? It means that if we're having two features of the same kind that come closer together, then this distance d, given by the wavelengths divided by this number, which is usually something like 3, it's not really possible to tell these features apart. And this number typically amounts to about 200, 250, 300 nanometers. And so any detail that is smaller than that, if molecules, vesicles, whatever that is, comes closer together than this distance of d, the representation in the image will be blurred. It will be not recognizable as a structure. Now, this equation can be found basically in any textbook of optics, of course, but also in textbooks of molecular and cell biology because of the enormous relevance of light microscopy to these fields. And the relevance is very clear. If you want to look inside a cell, a living cell perhaps, or even living tissues in 3 dimensions, there's no alternative to using a light microscope that just uses a regular lens and regularly focused light. Electron microscopy, of course, provides a higher spatial resolution. There's no doubt about that. But with electron microscopes, we cannot really see inside a tissue, for example, inside a cell in 3D without chopping it in slices if you want to go further down, and definitely not in a living one. Now, in the end, this is what people believed in the 20th century, and the gold standard towards the end of the 20th century became the so-called confocal microscope. And I think many of you will have heard about the confocal microscope. This is a microscope where you have a light beam that is focused across in the focal plane and then scanned across the specimen. And the confocal microscope provided advantages over the normal, say, epifluorescence light microscope, such as it gave a kind of 3-dimensional representation of the object if you, say, look deeper down into the cell, so you got a 3-dimensional image, and also it provided background suppression. However, in terms of resolution, the confocal microscope didn't change the game. So as a matter of fact, here we've recorded a sample, and you see, obviously, this is a 2-color or multi-color kind of recording, but we have no clue what it is. You say 250 nanometers, this is the resolution limit here in the confocal microscope. And the structural details that are in here are much finer. No way to see what it is. Now, the development of STED microscopy, which I'm going to talk about in a few minutes, has changed the notion about what the light microscope can really do. It showed that, if you will, there is physics in this world, or there is physical chemistry, or whatever you would like to call it. There is a way, a viable way in this world to overcome this barrier, get a much higher spatial resolution, but still maintain the condition of focusing. Just regular lenses and just focused visible light. Now, since we have developed this microscope, STED microscopy, of course we have the option of recording this feature here in the STED mode and see what it is. And here it is. So this is now recorded with a STED microscope, and obviously the spatial resolution is much higher than before. And then we can see what it is. So these are nuclear pore complexes from frog epithelial cell line. And because the resolution is increased by an order of magnitude, we can zoom in, and it makes sense to zoom in. And we see that these nuclear pore complexes have this 8-fold symmetry. You find, say, A molecular subunits. In here you see what it looks like, what is the range like. Because the spatial resolution here is of the order of about 20 nanometers in the red channel and about 30 nanometers in the green channel. This is what a confocal would look like. Well, you can compare it or not, but this is what we can do now. Now, some of you may think, okay, now they've done that in Göttingen, and of course they've developed this type of thing. Well, it works nicely, obviously, but it must be very complicated and in the end definitely also extremely expensive. it may be due to some important technological advancements. And the point I would like to make here, this would be an incorrect conception. This was, in fact, done with a student lab course setup. And if you ask me, the reason why this change has happened is not due to technological advancement. Of course, technological advancement helped. That's certainly true. Better lasers and so on helps. But the change, the reason for this change, is actually due to very simple and, of course, as we know now, important physical insight. And this is what I would like to convey to you. Being a physicist, of course, I always try to ask, what is the reason why something works? And so here the question is, what is the reason why we can take these images now, but say 20 years back, everyone said, no way, no way, you won't get better than what you've seen before. And so I will concentrate on the question why. Some of you may say, okay, I don't care about the why, I want to just apply it. But then you should keep in mind, if you want to apply it properly, if you really understand the potential of a methodology, it's critical that you really understand how it works and what the real basis is. Because only then you will understand how you can apply it, what the limitations will be, but also it will give you a feel for how this field is developing. And many of you will know that this field of, it's called now super resolution microscopy, is developing very quickly. There are all the new methods coming up with new acronyms and so on. And you don't know how this relates, how this compares. If you understand how it works, what really the gist of the whole story is, then you can put it in perspective, and then you can understand actually how you can make use of it. Of course, trying to explain how it works, I'm going to be very primitive in the way I'm explaining it. And it is very simple. You can explain it, I think, to everyone who has a basic understanding of natural sciences. Now let's assume this is the object. So what is the object? It could be, if you will, microtubules or something consisting of molecules. In any case, it's molecules. That's because nature is made of molecules. And we do fluorescence microscopy. For the moment, we're going to restrict ourselves to fluorescence microscopy, although if you think about the concept from the physics viewpoint, it's not limited to fluorescence imaging. But let's assume it's fluorescence imaging. That's the most important, say, part of, most important type of microscopy for the life sciences anyway at present. Now, in a fluorescence microscope, the molecules are labeled. So there is a dye placed on the molecule, or a GFP, so perhaps genetically encoded even. And then what happens is actually these molecules, these fluorescent molecules, they have two states, two basic states, a ground state here and an excited state. And if they absorb light, for example, green light, the molecule is raised from the ground state to the excited state, then the molecule comes down again, it relaxes again, but then it emits a photon, a light particle that is shifted in wavelengths to a longer wavelength, so from green to red. And this fluorescent light is actually what is being recorded. And this is how we see the molecule. So we don't see the actual proteins in here, we see the fluorophores. That's because that's what is made visible in here. Now, it has always been clear that you can separate features that are further away than the diffraction barrier, no doubt. Because they're further away, no problem. The problem of resolution exists only within a limited range. So we cannot separate this from this and this from that, simply because it's closer than the diffraction barrier. You see now that this is the typical barrier, the distance about 250 nanometers, or this is the diameter, and we cannot separate this from this, and this from this, and this from that. So why I'm saying this? I'm saying this for the following reason. If you want to find a solution for separating features at a very arbitrary resolution, it's enough to concentrate just on that region, or to say, okay, I'm just going there and find a solution for this region. Once I have found a solution just for this region, then separating the rest of the object is trivial, because the solution that I have found for this here can be instantly applied then, or applied later, to the rest of the object. And so when I'm explaining things, I will just concentrate on this region, and it's absolutely enough. If I find a way to separate this feature from that feature, that feature from that feature, you can believe me, the rest of the story is told because I am applying this solution to the rest of the object, and that's it. Okay, So we have to find a solution for separating this feature from that feature and that feature from that feature. And let's check if there is one. People have said there is none. Okay. Now, we find a solution. And one solution to the problem is the following. If you think about the problem, what is the problem? Why can't we separate this from that and this from that? One way of saying it is we cannot concentrate the light, the illumination light, any further than this blob, as I explained. The lens produces a spot of light, as we learned at school, and you cannot make it smaller than this value, than this distance, this spot of light. And because of that, all the features that fall within that region will be illuminated at the same time. More or less at the same time, because the light comes at the same time to a large area, not to a small area. Now, if we could concentrate the light further down than this, okay, there are, in principle, ways of doing that, but not with a lens. For example, with a very tiny tip, then we could go to this first, and to that first, and that first, and that first. Then we could separate it. But if we maintain the concept of focusing, using just lenses, and keep the light freely propagating, there is no way. All of them will be flooded at the same time. And so people have said, okay, forget it. You will not be able to separate anything. Now, if this is the problem, keep in mind that all of them are flooded and illuminated at the same time and give off light at the same time. Maybe there is a solution to the problem, namely, make sure that the molecules that are inevitably flooded here at the same time with illumination light, green light, are not capable of sending light back at the same time. Because if we manage to keep some of the molecules, for example, here dark, okay, despite the fact that we have, say, flooded them with light, with illumination light, we may be able to separate the bright ones from the dark ones. Or you can phrase it differently. While we can't change the fact that all of the molecules are flooded at the same time with illumination light or with excitation light, we may be able to change the fact that all the molecules that are flooded with that light at the same time are in the end capable of sending light back. Because if you manage to change that status of the molecule, able to send light back or not able to send light back, we may be able to separate those that can send the light back from those that can't send the light back. And this is exactly the idea of this STED microscope. Now, in the STED microscope, we use a beam of light on top of this beam that has only one purpose. Make sure that not all the molecules that are flooded here with light, the green light, excitation light, are in the end capable of also sending light back. Now, if you're trained as a physicist, and then you think, oh, how can I do that? The first thing that comes into your mind, if you think about it in that way, about a problem, is to use a very basic phenomenon, which is called stimulated emission. By the way, this phenomenon is used now for pointing here the beam onto the screen, but that's a different story. It's a very basic phenomenon. What does this mean? Now, usually molecules are excited from the ground state to the excited state because they are in the ground state. This is the only reason. Usually they are at room temperature in the ground state. But if the molecules are in the excited state, you can also just send them down by light, just as you can send them up. So there's no fundamental difference between sending molecules up or down. But if they are up, you can use a beam of light that just sends them down. In order to do that effectively, two conditions have to be fulfilled. First of all, the wavelengths of that, say, down-sending light, of this, it's called stimulating light, that's the technical term, has to be redshifted in wavelengths. So it has to have photon energy that is longer, because it should not excite molecules, it should de-excite molecules. And since the photon energy is lower, of course, then the molecules cannot be excited because the energy is not enough. But the molecules that are here in the fluorescent state can be instantly sent down to the ground state. So wavelength condition is the number one condition for keeping the molecule dark, so to speak, with this phenomenon of stimulated emission. There's another condition that needs to be fulfilled. Of course, you have to make sure that there is a red photon at the molecule in the case the molecule is excited. And that, say, condition is fulfilled by making sure that the beam here, the red beam, is bright enough. So if the red beam is bright enough, there's enough intensity of the red beam, then, of course, you can be sure there's always a red photon there that gets the molecule down in the case the molecule gets excited. So these are the two conditions. And we have to fulfill this condition One condition is, I said, the wavelength. The other one is the brightness of the beam, and this is shown in here. So if the red beam gets brighter than this value, which I put here as IS, then you can say the molecule is turned off. It's not capable of sending any light back to the detector, because as soon as it gets up, it's instantly sent down to the ground state. Okay, now we know how to turn molecules off, to keep them dark. Of course, it wouldn't make sense to keep all of them dark here. This would be nonsense. We see nothing. So we have to make a choice. We have to make a selection. And this is why we use, for off-switching, so use this beam in a patterned way. And the most simple thing you can imagine is, of course, to use a ring. Because this gives an increase in resolution, as you will appreciate, in all directions, and keeps molecules here more emissive or still bright at the center of the beam. And now, we have a better resolution because we can discriminate the molecules from here from those that are outside. So we don't see the strand here, and we don't see this part here, and this is not bad. Now, we would say, this is not bad, but we want to get better, of course. What do we have to do to see, for example, just those molecules here at the center? It's very simple. We have to turn them off. And I had mentioned the conditions for turning them off. One is the wavelengths. We keep that. The beam keeps the wavelengths. But the other condition is to make sure that there is enough red photons, so to speak, so these redshifted photons at the molecule, to make sure that if the molecule gets up, it gets down. So what do? We increase the intensity such that this condition here, being intensity larger than the threshold, is here present at a larger area. So we just crank up the beam, and then more molecules will be silenced, and only those molecules here are in the end of remaining in the excited state. And so we can separate those from those, because only those are capable of sending light back, despite the fact that all of them are flooded with green light. So we can't change that. All flooded with green light, but these are dark, and those are bright. And because they are different now in the capability of emitting light, we can separate them. We can separate these three molecules from the rest. Okay. How do we get the rest of it? We just got now the three ones. Well, we have to move this beam across this 200 nanometer zone. Now we see signal from here, measure the signal in a detector, put it in a pixel in a computer, and then we go to the next. And of course, we can separate them because they are forced to fluoresce sequentially in time, because we play this, as I'm calling the on-off game, so to speak. We play on-off with the molecules. And this is how we separate the features within this zone. Of course, we can adjust the resolution by, say, adjusting the intensity. Now fewer molecules are turned off. The resolution is only of that size. And now the resolution is poorer. It has advantages if the resolution is poorer as well, because then you don't have to make many fine steps. You can be quicker in your image if you want to image fast. And you don't need high spatial resolution. It's better not to turn off so many molecules and go quick over the sample. So you can turn your resolution, you can adjust your resolution and the speed of imaging at the same time. And they are, to some extent, mutually exclusive. That's inherent in the concept. Now, it's also clear that resolution cannot be governed by Abbe's equation anymore. So this D now, if you look at it, follows a different function. And this function is now given by the wavelength still. So this is the diffraction limited component. But there is this new term in here. I is, so to speak, the intensity that you have here at the peak of this ring-shaped beam or donut. And IS, of course, is this material constant. That is the intensity that is required to shut the molecule off. Okay? So that's it, basically. And so we have now this equation, and this equation tells us that we can overcome the diffraction barrier and get beyond. Now, if some of you will have a physics background, and they will say, okay, what is actually done here is a so-called nonlinear, transition, or if you call it a saturated transition, go from on to off, and you can call this a nonlinearity. That's correct. It's not wrong, but it doesn't get you to the point. So if you have, say, a physics background, and of course for those who don't have, if you want to really understand the point, and this is what I'm saying here, what we actually do, and that's the gist of the whole story. In fact, of the whole super-resolution field is we prepare the molecules in two different states. I indicated that already. We make sure that the molecules are in two different states, despite the fact that although they are very close together on a, say, diffraction-limited region. Here, they are forced to stay in the ground state, so we'll make a check which state they are, you will always find them here in the ground state. And here, they can assume the excited state. And this difference in states allows us to make the separation, despite the fact that all the molecules are treated in the same way by the excitation light. And this is how we can separate. This is a fundamental principle, very fundamental principle. Okay, and as I said, the rest of the molecules, how do we get? We just move the beams across that and then see the features sequentially in time, and we can separate it, at least in the cartoon. Anyway, now we're done with the zone. What happens with the rest of the object? Well, it's very simple. You can move the beam further. Why not? And that's a very easy way of doing it. And say, like commercial microscopes typically do that, and most, say, stat microscopes do that. But of course, if you just move the beam like that, it takes a lot of time if the area that you image is very large. And so the larger the area or the volume, the more time it will take. That's one option. And so you can say, oh, it will take a huge amount of time to get my picture if the area is very large. Is that inherent to the principle? Is that always like this? The answer is no. Because we could do the following. We could have another one here, and so on. Because, as I said, each beam here has to do the job only within the 200 nanometer zone. Here we can have another one, here we can have another one, and can have many detectors at the same time. For example, a CCD camera, for example, that records the fluorescence coming from here, and so on. At the same time, thread out for each scanning step at the same time. And this way we can parallelize the imaging process and be fast if you want to image large areas. Do we have to worry that this and this is there at the same time? No, because it's further away than Abbe's diffraction barrier. And if it's further away than Abbe's diffraction barrier, we can always separate. We can say it's sparse, and because it's sparse, there's no problem of separating it. So the problem exists only within this area. And as I said, once you found a solution for this, you do it for the rest at the same time, and that's it. Okay. So in many cases, the recordings are still done with the single beam. And if you look at scattering tissue, in the neurosciences, very often you have that. Then it's still advantageous, or in most cases advantageous, to use say, a single beam. Anyway, so some of you will have wondered how we make that ring-shaped beam. I'm not going to bother you with technical details, but it's not very difficult to make that. One uses a piece of glass that has a difference in thickness, and the difference in thickness imprints a phase change from 0 to 2 pi. And because of that, the opposing parts of the wavefront, if you put it here in the beam, will be out of phase by pi. And this is why you don't get, say, a blob of light here, you get a ring. But as I said, the ring is also diffraction limited, so that's not what gets smaller. What really happens here is that we turn molecules off because we do not allow them to assume the excited state. We force them to the ground state. And this is how we keep the region in which the molecules are allowed to emit, where they're allowed to assume the excited state, very small. Okay, now that has been how it works. A few examples. I'm taking random examples here. So this has been relatively early on. That was done with scientists who worked in Göttingen in those days, and he came to my lab and said, oh, I have discovered a protein. We gave him this interesting name, and I don't know what it looks like in space. Can you image that? And so we've done that. And you see here, it has been found instantly when taking the images that this protein forms this little basket when it arranges the structure, protein structure, and protein arrangement at the synapse. And without going into the details of this study, of course you can take multicolor recordings, as you can see here, labeling different terminals of the protein, and then you can arrange them in space. You take different views from different directions. So this is the confocal recording. Even after some mathematics, you don't get better than this. And this is what you have here in the stat microscope. And then you can come up with a three-dimensional model of how this protein is arranged in space. You see the numbers here, 40 nanometers, 50, 80, and so on. Keep in mind, the wavelengths that has been used here was 750 nanometers. So this is a fraction of the wavelengths. Still, you get beyond the diffraction barrier and see the nanoscale arrangement of proteins at the synapse fairly easily. Just push a button, get a picture, because the method per se gives the image instantly. This is another example that I like. This is a collaboration with a person working on HIV. Virology is, of course, a very important area of application because the viruses are typically between 30 and 150 nanometers in size, so just below the diffraction barrier, and there's no way to see something with a light microscope normally. And this person was interested in the distribution of a certain protein called ENF on the viral envelope here, on the membrane. They had no clue whether they are dispersed or not, so I asked, why don't you do electron microscopy? They said, oh, it's very hard to get the immunolabeling in electron microscopy because you don't get the stoichiometry right and all these problems. It's better to do a light microscopy. So we did it. Confocally, no way to see anything because the virus is smaller than the diffraction barrier. Then with that, we could see that this protein here forms these little structures in here, or these arrangements. And as it turned out, so if you correlate it, of course, with the size of the particle, it turns out that this distribution of protein depends on the maturation state of the HIV particle. So in other words, in here, if the HIV gets mature, it gets all its ENF in a single place, and this seems to be a condition for being for infectivity. So here, cut a long story short, STAT helped getting an important insight into the let's say steps of becoming infective as a HIV particle. Well, another advantage of this method, so some of you will, of course, will be interested. What is the particular advantage here of this method called STAT? Is that you get perfect colocalization, especially if you use a single donut beam, a single ring-shaped beam for turning off the molecules. And that's what we recommend. That's what we usually do. And my group also synthesizes dyes that are very well suitable for the laser wavelengths that are available now. And I'm sorry. And you can colocalize things very nicely because you can be sure that the fluorescence must come from this region. There's no way out because here it's turned off with a single beam. And if it is 2 dyes or even 3 dyes, it must come from this region. So you have a perfect colocalization. This is also important. And you can tune the spatial resolution. I'd like to give you some examples. I said there is an inherent trade-off between speed and resolution. That's fundamental because it's on-off. And the things that are turned off, they cannot give signals. So you have to turn them on at a later point in time. That means, obviously, you trade off speed against resolution. It's inherent. There is no way. But there's various ways of doing it, and still large room of getting, say, of balancing between the two. And you still can take fast recordings. So it's not that you're not able to take it if you do it properly. So this is a snapshot of vesicles in, you see it here, neuron in confocally, but now this is a STAT. And so we managed to see here individual, say, synaptic vesicles moving, so at 28 frames per second. So this is video rate. So this paper was important because it showed that you can go past the barrier and still have a very high speed and under sort of, say, lifestyle conditions in this case. This is another example. This is even faster. This is now 125 frames per second. So here it's 4 times, the recording is actually 4 times faster than the display. And this is neuropeptide vesicles moving along motor neurons done in a living Drosophila larva. Actually quite nice. So this is that on the green fluorescent protein. Now what happens if the molecules get very quick? And then you're not able to take movies anymore because they become quick, and there may be just a few of them. Is this the method useless? The answer is no. Because then you can do something that biophysicists call correlation spectroscopy, fluorescence correlation spectroscopy. You check out the molecular diffusion, you quantify it, and then you know how quickly a molecule diffuses. Now, confocally, people have done that extensively, no doubt. And this was important for many applications. But they have always been limited to measuring areas that are of that size, no way out. Here, now we can make them much smaller so that we see details of diffusion from much smaller areas, and this can be made down to about 15 nanometer resolution. So you can imagine a 15 nanometer spatial resolution of diffusion measurements in the plasma membrane of a cell. In this case, this was done with artificial bilayers, but in living cell we got down to about 20 nanometers. This is not the end. It's just to show you what can be done so far. Living neuronal tissue. Yeah, so this is taken out of a slice, so hippocampal organotypical slice, and you can follow the movement over time. So these are time-lapse recording with 20-second breaks. You see how this cup is changing, is forming after 180 seconds. So you see slight morphological changes going on. And of course, one can study now, say, the spine next. And, well, I think most of you know that this is connected, of course, with memory formation and so on. So I think we have provided a tool that helps us getting a much better understanding what's going on here at this scale for neurons. Of course, there is tissue also here and here, but there's only one neuron that is labeled. This is why we see this thing neuron in here. How deep can we go down? So if you want to go down deeper into the tissue, this is what has been done so far, 115 microns, without very sophisticated optical correction. So if somebody works hard on this problem to get better, I'm sure he will or she will get better. But this is can be done with relatively little effort to go down to about 115 microns, still keeping the resolution that is substantially better, 3 to 4 times better than the best confocal or two-photon microscope. And the structures here, the details here, the dendritic spines are of that order. So you can get meaningful information out of this. Now, say more spectacular, if you like, a bit more challenging, obviously, also, is to do it straight on the upper layer of the brain of a living mouse, anesthetized mouse. So this is a transgenic mouse. It expressed YFP cytosolically in some of its neurons. And you see now quite nicely the dendritic spines here. This is in the visual cortex, just 20 microns deep down. And this is a time sequential recording. So the mouse, of course, was fully controlled, and there was a tracheal cut here so that it doesn't breathe through the nose and things like that to make it still. And it's a time sequential recording, as I said, with 5 minutes time intervals. And you see there's slight morphological changes going on here. Apparently the brain has to change they have to change slightly their position. So you don't know what the mouse was thinking or dreaming or whatever. But there is morphological changes going on. And you need the high spatial resolution to see these little changes going on. This is over a period of minutes, so to speak. Now this is just cytosolically labeled. Again, the resolution is substantially higher than what you normally have. So we have tried also looking at actin in the fiber bundles. This is work that comes out soon. So it works. It gets you down to high spatial resolution. The level of detail that you have here is not bad. Again, this is the anesthetized mouse. Raw data, there is no mathematical treatment of the data. This is also important to understand. And here, so this first acting type of imaging, I think this will become better soon. But still, it shows that you can get a level of detail that is not bad, 60, 70 nanometers, even 40 microns deep down in the brain of the, deep down, so to speak, in the brain of the living mouse. So there is a lot of activity going on in this area, and there will be more of it. But it's important that we can access now the spines in the living mammal. One can imagine now doing experiments like giving cues, visual cues to the mouse, and then seeing what's going on. You can optically deprive, you can deprive the eye or something, and see how maybe the spines are formed and so on. So this is work which will come for sure. So this is an interesting area. Here again, if you do it in hippocampal slices, you have a higher clarity, no doubt. So it's still harder to do it in vivo. If you're just at the slices here, you can nicely see here the actin structure again in the living hippocampal slice. Okay, so with that, I'm coming back to the basics, and you've seen here all kind of numbers as far as the resolution is concerned. Something like you've seen 70, you've seen 30, you've seen 15. And so some of you will ask the question, what is the kind of resolution that we get with this method? Is it 15? Is it 70? Well, the answer to the question is, I should say, twofold. One of them is the conceptual limit. So what you can in principle get if everything is perfect, there's no aberrations, nothing else, and so on. And what is the practical limit that you get? Now, the conceptual limit is important to understand because this is going to stay. This will also be true in 30 years, most likely. The practical limit is something that is a snapshot of development right now. So 19th of December, 2013. And the conceptual, of course, as I said, is easy to answer. It's clear that if you shut off more molecules, then we will have a high spatial resolution, or more molecules on average, and we will have a high spatial resolution. And this is also reflected here in the equation. If you look at the equation, if I gets large over S, then D goes down to 0. So 0 means, oh, what does this mean? Well, 0 is not to be taken perfectly literally. 0 means the molecule, the size of the molecule, because that's the least separable unity that I have is a molecule. I'm separating by having the molecules in two different states. So this is the molecule here is the fluorescent state, here it's the dark state. And then I get signal from here, and then I can also see the neighboring molecule, although it is just at molecular distance, because it emits at a different time point. And so this is how I separate the molecules. So again, we can have molecular scale resolution, although we use regular objective lenses. That's the point. And normally propagating light, again, by having them in two different states at a time point of measurement. So this is, of course, a paradigmatic change in what people can now imagine that a light microscope is capable of doing. So then the question is, okay, if the molecule is the limit, what is the practical limit right now? Well, practical limits are, of course, you have to keep the minimum here, minimum. You have to keep it down to very small numbers, so 1% at least. Otherwise, you will also shut off the molecule, and you cannot separate the two. The number of state cycles, so how many on-offs you can do, that is also very critical, because once you turn the molecule off, of course, you want to turn on this one. But if it's not possible to turn it on, so if it's bleached down, you cannot get the signal. And so the number of on-off cycles is critical. And so with organic dyes, say, use a good stat microscope, you should anticipate 25 nanometers of spatial resolution. And this is quite a lot. This is a factor of 10 beyond the diffraction barrier. This is just an indication that you can go very far if this problem here is sorted out. So if you can turn it on and off many times, you can demonstrate. It's like say a pilot experiment to show you what is coming. There are so-called defects in diamonds. You can be turned on and off many, many, many times. There is no limitation. Because there's no photochemistry going on, there's no bleaching going on, and so you can turn them on and off. And so with those, we managed to show, say, areas of measurement down to 8 nanometers a couple of years back, 4 years back. And if you remove spherical aberration, I don't want to bother with details, you can go further down, so down to 4.2. The record is 2.4. And the reason why I'm showing this is to show you it's a development that is going on, and the physics gives it, so to speak. If you sort out the molecular states, then we will get there. There is no doubt about it. So the resolution problem is not a conceptual problem in physics anymore. It has become, if you will, to some extent, a chemistry problem, molecular state problem. And those guys who come up with the best molecules to get between two states, they will get the best images. I mean, there's no doubt. Okay. Now, with that, I'm coming to the last part of my talk, which is, so to speak, to give a general outlook of the development. And as I said, you will understand the whole development only if you understand the principle. And the principle behind this game of overcoming the diffraction barrier is this, what I'm calling the on-off principle. You've seen it. Turn things off and to see only that one, and then later on you turn the other one off and see the other one. And this on-off principle is in fact a principle of two states. What we do is we transiently get the molecules in two different states. And because they are in two different states, we can separate them. And once you realize that in full, then you know, oh, this is a very, very, very fundamental principle. And in my view, this has not been fully recognized. People in the past, especially physicists, they were, thinking about resolution in wave terms, but not in state terms. And because they thought more or less about wave terms, they were also trying to do something with the waves. But they forgot about the potential that is in the states. And this is my point. And instead, it's very clear. How does it work? I mean, we get things in two different states. Here, the molecules are forced to stay in the S0, here in the S1, and this is how we can separate them. And this instantly also tells you the weakness of this concept. I told you that you need a certain intensity, of course, to shut the molecules off, to keep them here down in the ground state, something like megawatt per square centimeter. It's not too large. You've seen it in the live cell, you've seen it in the living mouse. But of course, it doesn't offer you room to get further up in intensity, because then at some point it will become too large. But why do we need these intensities here? It's very simple. Because the lifetime of the state here, of this state, is just 1 nanosecond. So this difference in states that we critically need here to separate the features is there only for about a nanosecond. And it means we have to hurry up to create a difference in states and register that, otherwise the difference is gone. And so this is why we have to put in light so quickly, means high intensity, because we need to set up this difference in states within a nanosecond. It's not going to stay there for longer. Now, once you realize that, and I realized that very early on, of course, then you know, oh, this is about on-off for 2 states. It's not critically stimulated emission. Stimulated emission is just one method of doing it in that way. So there must be other pairs of states in the molecules. If you look at molecules that have many states, that could do the same job. And so one option is, and those who have a chemistry background know about that, there are also dark states, so-called triplet states or other dark states. And of course, you can push a molecule to some extent also to those dark states in various ways. And if they stay in a dark state, they stay there for longer because the transition from here to here is not allowed as a dipole transition, as physicists would say. And then it stays there for about a microsecond. And then the difference in states is maintained for longer. Okay? You pump it there. Here you have it in the bright state, here in the dark state. But this difference in states is now for a microsecond. It means the intensity requirement to shut it off or to get it from one state to the other is reduced by orders of magnitude. By 3 orders of magnitude. Nanosecond, microsecond, 3 orders of magnitude. Again, you flood everything with light, but only this one is allowed to emit. This is dark, and this is how you separate it. Okay. I didn't start out with this because I was scared with photobleaching. If you go triplet states, I don't want to bother you with that. But another option is to do a cis-trans isomerization. Some molecules, they can make some, say, flip an atom or so, and they can go from cis to trans, and trans is dark and cis is bright, and the lifetime of the state is relatively long. So milliseconds to seconds in this case means you further reduce the intensity, and then you shut them off. And then you get the molecules here off, and here you get them on. And so these difference in states allow you to tell them apart at much, much, much lower light levels. And so I couldn't have called it STAT. I had to give a different name, so I gave this name, resolved. STAT stands for stimulated emission, and there's no stimulated emission in it. Okay. Now, there's even for the switchable fluorescent protein that can go from one state to the other. So I was excited about that because it means you have genetically encoded marker, and then you go at low light levels, and great. But when we developed this, and we started out in 2003, we realized it's not very easy. You have to find fluorescent protein that can be switched many times between on and off. And that turned out not to be very easy, so we had to work on the development of fluorescent proteins. And it took us some time to get them, most of it random mutagenesis. And in the end, we got them. And so this has allowed us to do a method that works like that, but at much lower light levels, and gives more room for improvement, of course, of resolution. And I'm showing you this now. So this, again, hippocampal organotypical slice is actin in this case. Again, high resolution, 2 hours continuous scanning, no visible photo damage. Again, this is important to show that this is a development, and it depends, of course, on the set of states that are used and the state transitions that determines, in the end, say, the practical, say, conditions for intensity, wavelengths, and so on. And it's not an intensity game, the breaking of diffraction barrier. It's a state game. That's important. Okay, now if you have low light levels, and so we have much lower light levels in here than in the normal STAT mode, you can say, okay, then we can use large area recording. Take a very large area. And how can you do that? You can do stripes. People have done that. They call it structure elimination. In my view, that is not going to lead anywhere for very fundamental reasons. Because you have to turn the stripes also to get resolution also in that direction. And usually, if you have just stripes, you get resolution in one single direction. So I thought, okay, why not crossing the stripes? Just cross them like that. And then you have here holes. And here the molecules are allowed to emit. Here they are not. But if you just cross stripes, you may think, okay, this is not getting round here. This is like a quadratic thing. And this is not good if you want to have isotropic spatial resolution here in this direction, the same resolution as in this and in this. And then if you think about it, then you realize that the resolution gain scales with the square root, inversely with the square root. And this really helps you, in fact, most naturally, to get here a round area of on-state. Why? Because here you have twice the intensity because you cross the light like that. But the distance is by square root of 2 further away because of the triangle here, you see. And so, okay, this is just compensated. And in the end, okay, the gist of the story is it becomes round. You don't have to turn the pattern, nothing. You just cross two lines, and then you have, say, a round hole in which the molecule is allowed to assume, in this case, the cis state, which is bright. You scan it like that, record it with the camera. You make a few scanning steps, maybe 10 or 15 in this direction, 10 or 15 in that direction. You have a huge field of view, and you're done. And here it is. This is normal wide field recording. Now this is done here. With 1 second, 120 by 100 microns living cell, because it was recorded with about 100,000 holes at the same time. So this is another example. 3-second recordings, 120 by 100, because it was recorded in a highly parallelized mode. And so again, just to give you a notion, you don't need just a single scanning point, you can have many. Now, so this is the story, so to speak, of the stat, and they have different names, or resolved, or whatever you like to call it. And the hallmark of these concepts is that you play the state game and you induce the state transition with a beam of light. So in all the methods that I've shown so far, we've used a beam of light, like the blue beam, like a donut-shaped or cross pattern or whatever, to determine at which position the molecule is on and where it's off. Okay? And so this is the hallmark of this. It has advantages and disadvantages. I will talk about in a minute. Now, in 2006, so in the interim, after we suggested this, an alternative method came up, which has become very successful. It runs under the acronyms PALM or STORM. This is the same thing. And here, this on-off game is played in a different way. Again, the same transitions are used in this initial suggestion, and the on-off game is played randomly in space. So initially, all the molecules are off, say in the trans state, then you turn them to the bright state, the cis state, with a weak beam of light that is not focused, but at some point somewhere a molecule will go to the on state. You see it goes to the on state, and if it goes at some point within this 200 nanometer zone, of course you have no clue where it happens, no idea. So what you have to do is you have to find out where it is located. Now you can do that under one condition, this is the restriction here of course, that this molecule is capable of emitting many photons in a row, in a bunch. Because then, of course, you can find out where it's located just by making the centroid of this, say, pattern of diffraction that is produced on a camera. So what you do, you take a camera as a reference coordinate, image it on the camera, calculate the centroid, then you know where the molecule went to the on state, register that point, and turn it off. Then you go to the next, do the same game. It turns on, calculate the position, turn it off. Again, everything flooded with light. But only this one is in the on state, the rest is in the off state. This is how you make the separation. The coordinate you get from the molecules, from the photons that come out. And this is how you do it. Again, the separation on-off, but you do the on-off individually, molecule by molecule. Here you do it in groups usually because you can determine the position. Each one has weaknesses and strengths. Here you need many on-off cycles because you turn the molecule deliberately on and off many times. Here, one on-off cycle is enough because once you've registered a molecule, you turn it off and that's it. But you need many emission cycles. You need very bright molecules. Here, it's not needed, fundamentally. So you need not so bright molecules, but many on-offs. Here, you need very bright molecules, and one cycle is enough. Of course, this is also tentatively slower because here, this on-off principle has got to the extreme, down to the single molecule level. So it takes a lot of time, of course, to get all the molecules, a representative number of molecules. Now, these people saw that you need this cis-trans ionization, but knowing the principle, of course, we suggested to use also other transitions like this dark state transition, and this has become a different name, GSDIM. We're going to talk on the details. You can elegantly parallelize this stochastic method because you can see large areas more or less instantaneously. And so it clearly has advantages. Gives nice pictures, as you can see in here. Beautiful application must be shown. Recently, this is by the Zhuang group at Harvard. In fixed cells, we can do that with live cells right now. And in this case, it's done with Stefan confirming this. And Eli, who is actually here in the audience, was heavily involved in these recordings. Again, here, of course, you need many photons to determine the position. This is an insight. So in that sense, of course, you need always many photons to get a position, just as you need many photons to come out, in this case, to get a position. Positioning always requires many photons, because one photon goes astray. If you have just one photon, it goes here, goes here, goes here. If a photon comes out, it could have come from here, here, or here. To get positions, you need many photons. Positioning is always done with many photons. That's very clear. But positioning is not resolution. You should never forget that. You cannot break the diffraction barrier by getting the position, by localizing molecules. No way. People did that in the 70s, 80s. They localized individual molecules. They followed that trace. Couldn't get an image because they get just the position. What you really have to do to get an image is you have to separate features. That was the problem. Tell it apart. It's closer than the diffraction may tell it apart. But the essential element for separation is, of course, the state transition. You have an on and an off state, and this is how you make it distinguishable, and this is how you make it separable. And by transiently placing the molecules in two different states, either here in this coordinate, coordinate targeted manner with the beam of light, or here in the stochastic manner, you make it separable. And you can use all kinds of state transitions in order to make it separable. So the whole story can be boiled down to one thing. What we do is actually, we break the diffraction barrier by transiently placing the molecules in two different states, state A and a state B. And then there can be many sets of states, one can imagine, not just the one that we have used so far. Maybe you have your own idea or whatever, to come up with a pair of states that is viable. And of course, the selection of states will determine the details of the performance of the microscope or of the imaging system. And this is, of course, very critical. But it's not necessarily tied to fluorescence or so. You can even use bound or a bound state and diffusing state as a kind of state thing. Okay, now with that I'm coming to my last slide. Obviously we can get better pictures, as you can see here, so this was the limit, and now we can separate this from that. Here we couldn't, and of course we can expand this equation, not a big deal, and here it goes down now to very small numbers. You can see here 0, which means actually the size of a molecule. Okay, a home take message. Why can we separate this strand from that strand? It's very simple. Because at the time, the molecules of this strand were in the S0, so off. Here they were in the S1, and vice versa. And this is why we can separate them here. They were all in the same state, and this is why we couldn't separate them. So the diffraction barrier, and in the end, is broken. It's not circumvented. It's truly broken. It's truly gone. By preparing the molecules transiently in two different states so that you can distinguish them. And this is what, in my view, was forgotten. People were just focused on waves. They thought they have to change the waves, putting in a needle or tip or anything, or maybe design special materials together. The waves behave differently. My take is let the waves go and do what they want. Let them propagate freely. Why violate the propagation of waves? Let the waves go out. Do the separation by the states. And that's the point. Resolution is not just about waves. It's waves and states. And so the question of the 21st century when it comes to resolution will be not only give me the best lens, does the best waves treatment, the best focusing, but the question is which states in the sample give me the best spatial resolution. And this is what future generations will ask. They will see the sample as a part of the image formation. The molecules that they look at is a part of the image formation. And if they can get them in two different states in a short period of time, the diffraction doesn't matter, and you can get the crispest images just by using focused visible light. Thank you very much.